All right. Good morning, everybody. Morning. Good to see you guys. Well, today we're going to dive back into the nervous system. So today we'll do a bit of a recap on a couple things, including this stuff, the neurotransmitters, then we'll dive into reflexes a little bit deeper, and then the autonomic nervous system. And I made another poll everywhere. Try it again. I think it will go better this time, but for the end, for a bunch of questions about the autonomic nervous system. So, yeah, so that's what we got. Any questions about anything moving forward before we start? Go ahead.
There's like hundreds of thousand controls are being generated as per they reach the targeted cell. So is the pain intensity, or like let's say the pain scale, is it related to how many of those controls are reaching?
That's a great question. Yeah. Yeah. So that kind of gets to the idea that the nervous system overall is frequency modulated, right? So when we'll talk about the endocrine system next, the endocrine system is more what we would call amplitude modulated. So with action potentials, there's only one height of those waves, right? Like we really only depolarize to the same height each one. So there's not like a big action potential or a small action potential. And so when that signal is going up to the brain from a pain receptor, it doesn't matter how tall the action potentials are because they're all the same height, right? So that would be the amplitude part. The way the brain interprets a little bit of pain versus a lot of pain would be the number of action potentials arriving from that pain receptor. Yeah, it's the same thing with a touch receptor, right? If we're touching ourselves lightly versus pushing harder, right? The only way the brain knows the difference is because of the frequency of action potentials produced by that touch receptor, right? And so yeah, that's a great question. Yeah, good. Okay. Yeah, well, let's just kind of do a quick recap here on two things. So you can go ahead and fill this out to kind of just touch back on what we talked about last time. And then this one too, we got into the neurotransmitters a bit with the poll everywhere questions. So just go ahead and fill this in as well. So when you go back to that table again, try to find each of these kind of diseases or drugs related there. I guess those are all diseases. Yeah, I guess, well, there's drugs there with the opioid-based drugs. And then just kind of which neurotransmitters related to those. And then, yeah, if whatever information you have from that table on how are those connections made. Okay, so let's spend some time doing that with the people on your team, then we'll come back and put all that together.
Yeah.
Kind of review here. These are so things that I wrote in here as far as which ion flows up here. So we got our sodium or calcium influx for those EPSPs, right? We've got our chloride influx or potassium efflux for the IPSPs. The neurotransmitters that tend to be inhibitory, glycine and GABA tend to be ones, even though I believe it's GABA that can go either way, right? Am I remembering that right? Or is it glycine? You guys got the table up? Glycine, thank you. Yeah, glycine can go either way, but we do find it in a lot of inhibitory synapses. GABA would be kind of our primary inhibitory one, and then endorphins and enkephalins are inhibitory, because we're inhibiting the pain signal going up, right? Glutamate's our primary excitatory neurotransmitter. Substance P is also always excitatory. Those two, just a simple look at those two pathophys ideas here. Botulism, ends up blocking the release of acetylcholine, right? We talked about those different docking mechanisms that are kind of messed up with the vesicle transport and docking and release with that. And then tetanus, remember, goes into the motor neuron, works its way up to some inhibitory interneurons, right? And then blocks those. So we're blocking the inhibition. So we end up with kind of too much contraction because we're not inhibiting the motor neuron like we're supposed to. So questions on that or what I wrote? Yeah, Deja? This one down here in the bottom? Yeah, I just wrote blocks inhibit because I ran out of room. Blocks inhibition of motor neuron is what it should say. Other ones? Okay. All right. So then when we look at the more at the neurotransmitter table there, a few other kind of pathologies or drugs related here. So Parkinson's disease, we talked about this one yesterday. In the substantia nigra in the brain, we have dopaminergic neurons there. And in that part of the brain, dopamine is inhibitory. Okay. And so we start losing those neurons in Parkinson's. So we lose the amount of dopamine released. So we lose inhibition, right? So we end up having kind of these jerky muscle contractions, too much muscle contraction, right? The two on the table that dealt with depression, serotonin and norepinephrine. Serotonin, we've got those SSRIs, right? Serotonin, selective serotonin reuptake inhibitors, right? So we're blocking the reuptake, leaving serotonin out there in the synaptic cleft longer. So have more of those calming and feelings of well-being effects, right? Norepinephrine is a tricyclic antidepressants act on the norepinephrine pathway, also reuptake inhibitors as well. Okay, then we've got our pain killers, right? Our opioid-based drugs. All of those act in shared pathways with endorphins and enkephalins, which are just blocking that pain signal from getting up to from the spinal cord up to the brain. So we're not aware of it. Myasthenia gravis and curare. Myasthenia gravis is an autoimmune disorder that destroys our acetylcholine receptors. Curare is the chemical found on poison dart frogs in the Amazon and places like that. If you get too much on your skin, then you die. You stop breathing. Really, it's a super powerful chemical, but that has been turned into a synthetically created drug that is also used in the lethal injection cocktail. If it gets into your diaphragm doesn't contract, right? And you can't breathe. So it blocks acetylcholine binding, right? It competitively binds to those acetylcholine receptors and doesn't allow for acetylcholine to bind. The one on there for anxiety was related to GABA, and this is benzodiazepine drugs that can treat depression. I'm sorry, anxiety. Yeah, go ahead. Yeah, that's it. Blocks ACH binding.
Yeah, what's above?
Above that one. That's related to myasthenia gravis, which is autoimmune, and it breaks down or destroys acetylcholine receptors in the neuromuscular junction. Other ones? Yeah, go ahead. After endorphins, enkephalin. They're kind of like combined together, have similar effects. Yeah. I know, my handwriting's so bad. It's really hard for me to write on the iPad. It's not that much better on pen and paper, to be honest, but it is a little bit better. Okay, go ahead.
Was this the inhibits link signal?
Pain signal. Yeah, yeah. Thank you. Yeah, go ahead.
That would be also for nerve.
For nerve pain. Yeah, I think I have seen the use of some of those drugs for that. Trying to think of the other one. I think it's called Neurontin, which is for nerve pain and depression too, which is interesting. Only reason I know that is because my wife's a psych MP. I don't I don't really know any of the stuff much about drugs beyond the ones that I've looked up, to talk about in this class. But yeah.
on the spine?
Mostly in the spinal cord. Yeah, yeah, for the most part there in the spinal cord because we're blocking that signal before it even gets up to the brain. Yeah. Other questions? Yeah, go ahead.
Under expression, and then how are they connected? Reuptake blockers.
Yeah, and then SSRIs, so those selective serotonin reuptake inhibitors. Tricyclic antidepressants are the ones that act on the norepinephrine pathway. Yeah, for sure. Other questions? Okay, good. All right. Well, let's yeah, let's move on. I do feel this doesn't apply to you guys too much, but honestly, for the past 4 years, I've just given that table and just said know it, and so I'm feeling better about the neurotransmitters for you guys this year. So just feel good about that. I do, but so all right, okay. Let's talk about reflexes. So when we talk about reflexes, we're the kind of general pathway for reflexes is shown here, okay? And this is just a good thing to kind of have in your mind of what's going on with reflexes. So there's always some kind of sensory receptor or sense organ that's going to be sensing whatever the thing that's a potential threat to us, right? That's what reflexes are all about, is saving us or protecting us from something that's a threat to our bodies, right? So we have some kind of specialized sensory organ or receptor that's going to sense that threat, right? That's going to create a little generator potential, which would be an EPSP, right? And that local potential is eventually going to create action potentials on the afferent neuron that it's connected to, right? That afferent neuron is going to send the signal up into a synapse in the spinal cord, most likely. And then we're going to then have a series of either EPSPs or IPSPs. And then we're going to have some action potentials on an efferent neuron. And then that's going to get down to some sort of muscle. And we're going to have contraction or inhibition of that muscle. Okay. So that's just kind of the general thing, right? Reflexes can be monosynaptic, which means we just have this one synapse here. Okay. Or a lot of times they're polysynaptic, where we have one synapse there, but then this would also branch to a different neuron over here and have a different effect there. Then we would have a polysynaptic reflex. Okay. As I mentioned last time, the average motor neuron has somewhere around 10,000 of those synaptic knobs. Okay. Other places where axons of other neurons are interacting with it before it gets down to the muscle itself. So just an incredible amount of inhibition and activation and modulation of its signal before it actually gets to our skeletal muscle. Okay, go ahead. Yeah. I mean, yeah, to be able to recognize that the small ones would be, those would be graded potentials, right? And then the big ones are action potentials. And so typically we've got, until we're on the axon itself, we're going to have graded potentials, right? So we've got graded potentials here around this sensory receptor that are eventually going to create an action potential on the axon, right? And then we've got graded potentials here on the dendrites and body of this receiving neuron until we get enough to then create an action potential on the axon. So that's the way to think about it. Okay, good question. Other questions? Yeah, go ahead, Brittany.
Clarifying for polysynaptic.
Yeah.
What's the end of this, so the brain neuron, that sense organ, like, is it connecting to that, like an intermediate synapse?
Yeah, this is just kind of general. We'll see a couple examples here in a sec that I think will help. We'll see some polysynaptic ones in just a sec. So, yeah. Okay, so let's look at two specific ones here. We'll talk about three actually, talk about the one that involves the muscle spindle, one that involves the Golgi tendon organ, and then we'll talk about one involving pain receptors. So the muscle spindles are pretty unique little sensory receptors that are found in our muscles. kind of zoom in on them. They're just kind of embedded right in there with our muscle fibers, okay, with our contractile muscle fibers. And we have kind of different portions to it. So we have the intrafusal fibers and the extrafusal fibers, okay? Extrafusal fibers are really just regular muscle fibers, and those are going to be kind of around them. But then these intrafusal fibers of these muscle spindles, they have contractile ends. Right, so we can see the striated part, the parts with the lines in them on the ends. Those parts can contract and change length. That's kind of like the way that we can kind of modulate our reflexes. Okay, we can as we learn to, for example, as a baby is learning to sit up or learning their posture. Um, Early on, right? You try to set a baby up on to sit up and just kind of falls over, right? And a little bit later, like as they're starting to kind of learn how to sit up, they start to fall over a little bit and they catch themselves, right? It's because they've learned how to send the right amount of signal to these muscle spindles to tighten these up just a little bit, so they'll be a little bit more responsive to that stretch, right? So when they start leaning over a little bit, the muscles on this side are telling the spinal cord, oh, those are getting too long. If we want to stay up, we need to contract a little bit, right? And so that's kind of a real basic way to look at it. But that the kind of sensitivity of these receptors can be modulated by how much tone we have on the ends of those receptors. Yeah, comfort. What were you gonna say? The last slide, sure. Yeah.
So the gradient potential.
Yeah.
Is that what a P wave would be on a PQRS?
It looks like a similar bump there, right?
Yeah,
But
because QRS would be
yeah, it does look similar, but no, unfortunately not. Yeah, yeah. The P wave would be a bunch of action potentials that went through the atria, right? And the QRS is a bunch of action potentials that go through the ventricles. Yeah, but it does look like that for sure. Yeah. Yeah, so we've got that. So we've got the part that can contract on the outside, right? We've got these big, large diameter myelinated neurons, like we talked about last time, that send signals super fast that connect to these. Okay, and we also have some efferent neurons for the ends. Those are the ones that change the tension on the muscle spindle. Okay, so there's pretty much two things that these sense. There's the static portion of the receptor. Okay, and we can see that right here. So it's kind of this part in the middle, and then there's a dynamic portion that's a little bit further out. Okay, the static portion is sending signals to the central nervous system about the length of the muscle. Okay, and then the dynamic portion is sending signals about the speed of movements, mostly stretch, right? So this one is more complex than the Golgi tendon organ that we'll talk about in a second, because it adds this component. It has two different things it's measuring. It measures length, so it's constantly sending signals up to our cerebellum about the length of all of our muscles, right? And this is how we stay upright. This is how you guys are in the posture you're in right now, right? Is because the cerebellum is taking all that information about the length of all your postural muscles and kind of fine tuning that signal that it sends back down to create the right muscle tone, to keep your head upright, to keep your back upright, right? All of those things, right? And that's all what a baby is learning to do properly as they learn to sit and walk and stand, right? So that's kind of the length part, right? That's kind of there, just maintaining muscle tone. But then the dynamic part is about the stretch that you get when you get the reflex, right? And that's a little bit more of kind of the potentially threatening part to our body, right? When our muscle stretches too rapidly and too far. Okay. So what does a stretch reflex look like, right? So if we already kind of looked at it in general, but now we'll fill in some details of different synapses, EPSPs, IPSPs. We'll fill in the neurotransmitters here, okay? And so we can start with the stimulus, okay? And you can fill in what that stimulus would be. We're really just taking a reflex hammer, hitting the patellar tendon there. That's gonna pull on the quads a little bit, right? So yeah, just kind of walk through this. If you don't know the neurotransmitters, that's fine. I think you'll be able to fill in a couple of them. But what would that stimulus be? What would we call that? What's the receptor we're using? There's a monosynaptic portion, right? That's the part that goes right here, straight to the motor neuron, right? And then there's a polysynaptic portion that goes, has an interneuron in between, right? So there's multiple synapses there. So that's the polysynaptic part, okay? So just walk through it just a sec with the people next to you and see if you can kind of figure out most of this, and I'll help you fill in whatever details you're not sure on, okay? Go ahead.
I'm gonna read this here. So this would be excitatory, this first. So, and they're
And you want to write in whether you think it's an EPSP caused in certain places or an IPSP. That would be the other thing to write in here.
Yeah.
All right, let's go ahead and come back together here. Most of you are working through that. The stimulus here that we start with, what did you call it? Stretch, that's it. Yeah, it's stretch. Okay, so stretch. The thing that's different about stretch versus what we'll talk about next in the inverse stretch reflex, we'll talk about tension being the stimulus in that one. The difference with stretch is it has this component of length to it as well, right? With a Golgi tendon organ, we're really just like, it's like how hard are we pulling on the tendon, right? With the stretch reflex, we could stretch it here. We could stretch it here, right? Depending on how long it is, we can still stretch, right? With the Golgi tendon organ, we have to stretch that tendon out all the way to get any tension on it. Right? But with this, since those fibers on the outside kind of tighten down as the muscle shortens, right? It can kind of get stretched rapidly at any point, which is just makes it a little bit different. So we'll use that term stretch to kind of define what that is, okay? The receptor, what is it? Monosynaptic. So the monosynaptic part with this, right? Uh, and it's going to generate that greater potential by using glutamate. Okay, so our first neurotransmitter here is glutamate. Is that the right one? Yeah. Okay, so glutamate is going to be our primary excitatory neurotransmitter. Okay, you know what? That's not the right spot. Sorry, that's not the right spot. I'm sorry. Always. Oops. Getting confused by where my arrows go. Yeah, so we get Yeah, so we get that generator potential by the stretch of the muscle spindle, which sends an action potential down here. The first one we need to talk about is this one right here. Yeah, that's the first one we need to talk about. That afferent neuron is going to release glutamate right there. Okay. And then what type of signal would that send? An EPSP onto the motor neuron for the quads. Okay. So we're going to say the afferent neuron releases glutamate, which causes an EPSP on the motor neuron or the quads, right? Which there's a bunch of them, right? This is simplified, obviously. Motor neuron for the quads. And then we're going to have an action potential that goes down that motor neuron, right? And then it's going to release a neurotransmitter at the quads. Which one would that be? That one you guys know? That's acetylcholine, yeah. All right, so that's where our acetylcholine release happens, and we get contraction. All right, the opposite of stretch would be contraction. So we're taking that potentially threatening stretch of the muscle, and we're trying to reverse it by shortening the muscle and making it contract. Okay, so that's the monosynaptic part. So this would be acetylcholine up here. Okay, and then the polysynaptic part. So the polysynaptic part, we're still going to start with the afferent neuron doing its thing, just like it did. Okay, it's also going to release glutamate, which creates an EPSP, but it creates that on an inhibitory interneuron. And then that inhibitory interneuron is going to release glycine. And what would that glycine cause? IPSP, yep, exactly, IPSP. All right, so we've got our glutamate here. All right, we've got our glycine here. Okay, and so then in the end, we get inhibition of the hamstrings. Really, it's inhibition of the antagonist of the muscle that was stretched, right? Because if we contracted the hamstrings, we would stretch the quads more, right? And that's what we're trying to reverse. Okay, so it's kind of tricky because it's like we have to activate an inhibitory interneuron in order to let it do its job of inhibiting the next one, right? Which makes sense because it has to release its neurotransmitter to cause inhibition, right? Okay. Questions on that? Yeah.
When we look at this figure, should we start at 10 from here?
Yeah, start at the muscle spindle. Well, you can start at the reflex hammer hitting the tendon, right? Which that pulls on the muscle, right? Which pulls on the muscle spindle. That eventually creates an action potential on that afferent neuron going back to the spinal cord, right? And then just go from there, right? Then you can do the monosynaptic part first that goes back and causes contraction. Do the polysynaptic part next that causes the inhibition. Yeah, good question. Other questions? Yeah.
Does the same pathway happen if like just doing a regular exercise like a knee extension? Do you get inhibition on the hamstring, or is that hamstring just not involved and it's just remains relaxed?
Yeah, I mean, it happens all the time, right? So if we're thinking about throwing a ball, for example, like we're going to stretch, right? We're stretching, stretching, stretching. At some point, that stretch is going to then switch to contraction, and that reflex helps us go from the loading to the exploding phase of any kind of action like that. Any kind of plyometric activity would be the same thing, right? We're going down, we're loading, we're stretching, and then the reflex kicks in, and that helps us jump back up, right? So it's really happening with most movements where there's some kind of stretch before we activate. Yeah. Great question. Yeah. It's all together. Yeah, because it's because this neuron activates them both at the same time. Mm-hmm. Good question. Other questions? Yeah.
So the
Yeah.
muscle spindle, that's where that's where the generator potential starts.
Muscle spindle starts the generator potential, which will turn into an action potential on the axon of that sensory neurons. Yeah. Mm-hmm. Good. Other questions? Okay, so then if we look at the next one, the inverse stretch reflex, okay? So this now involves this other sensory organ called the Golgi tendon organ, right? So there are these little sensory receptors embedded into our tendons that have the purpose of detecting tension, right? So we would call the stimulus here tension, which is really just how hard we're pulling on both sides of that tendon, okay? These are commonly abbreviated as our GTOs, Golgi tendon organs. Okay, so now instead of stretch being the thing that starts this with the reflex hammer, it's contraction, right? So number one here is the Golgi tendon organ is sensing excessive tension, but we're doing that because we're contracting our quads here, right? So this would be like doing a knee extension, right? Like we're pushing out, trying to contract our quads as much as we can. Okay, so when we activate that Golgi tendon organ, okay, so GTO activated, we get an action potential on that sensory neuron, right? And then we've pretty much got a similar situation. We get back into the spinal cord. And we're going to activate an inhibitory interneuron here. That's the red one. Okay. So that red one you can see connects to the dotted green line, right? That dotted green line, when it's dotted like that or dashed, it just means that it's inhibited, right? So that the easiest way to do this, because there's not a ton of room to write on this slide, would be everywhere we see the plus signs, it's going to be glutamate. Okay, where we see those minus signs, negative signs, it's going to be glycine. Okay, so it's just pretty much the reverse of the last one, right? Where we've got glutamate release here. So we get an EPSP on this interneuron that releases glycine here, and we inhibit that with an IPSP there, right? The other part is glutamate here, which activates glutamate again, the more glutamate release here, which activates this one, and then we go down and we would get, that would be the only one that's not glutamate for the positive, right? This would be acetylcholine there, just like it is in all neuromuscular junctions. Okay, so we're now inhibiting the contraction of the quads, and we're contracting the hamstrings. All right, and so then this could be applied to a throw or a jump as well, right? So we stretched back to stretch these muscles, and then as we go to contract forward, the muscle spindle helps us do that. But now we're contracting, right? At some point, the Golgi tendon organ is going to kick in and cause all of our muscles that are causing internal rotation and adduction to relax, right? And then that activates the ones back here. They're going to slow down our arm, right? So we can slow down. We can do those eccentric contractions to slow down our arm, right? So both these reflexes are happening with all types of movements like that. Okay, questions on that one? Okay, so then our last one here would be the withdrawal reflex. Okay, and so the withdrawal reflex is using a pain stimulus, right? So pain would be our primary stimulus here. This would be a nociceptor. Okay, so we get pain, and it's also known as the flexor withdrawal reflex too. And it's really because it activates all of our flexor muscles at the same time, which is kind of interesting when you think about it, right? Our elbow flexor, right? In this example, our finger flexors, our wrist flexors, that kind of shows it going back. But for the most part, it's going to go like this. When we step on something sharp, like walking on the beach, when we step on something sharp, right? What do? We flex our hip, we flex our knee, we dorsiflex our foot, right? All those things, we're just kind of pulling away from it, right? And so, yeah, so it's the same pretty much idea here where we're getting that pain signal, action potential goes back, we would get glutamate release right there, right? And then obviously at all the muscles that get activated, we're going to get acetylcholine release. This is just showing one muscle, right? But it's activating multiple muscles to pull you away from the danger. Okay. Questions on that one? Bless you. Yeah. Yeah, so a lot of them are polysynaptic, even though it's activation as well. And so, yeah, you saw that on the Golgi tendon organ one here too. The image shows an interneuron in both cases, even though it's excitatory. Yeah, good, good catch. So, other questions on that? Yeah. The uh, spot where it's not is with this one, with the activation on the muscle spindle, but all the other ones, from what I've seen, show an interneuron. Yeah, yeah. Okay. Other questions on that? Go ahead, Sam.
For the withdrawal reflex,
Yeah.
would we also see the similar pattern of like inhibiting muscles that would bend into?
Yeah, I would guess you probably would. The yeah, it doesn't show it in this example in this image that I found, and I didn't really see it in the book either, the book for this class. But I'd be surprised if there wasn't inhibition of the antagonist muscles, because that would be how you would ensure that that the joint would move in the right direction. But yeah, yeah. Go ahead.
Is glutamate also released by the interneuron as well?
Yeah, it is. Yeah, yeah, right here as well. Yeah. Okay, great questions. One thing I just forgot to say, the quizzes each week should always be due by Monday. I just had that when I went on the grade book yesterday, I saw that a bunch of you hadn't taken the quiz yet. I was like, oh no, everybody forgot to do the quiz. It was because they still had to do on Tuesday from last year's class. So that's definitely better to have all that done beforehand, because a lot of that information you'll get with the video and the quiz will be helpful on both days of class for the week. So I'll go back in and make sure all those due dates are right. But just so you know, that is the plan, is to try to have those due on Monday before class. So okay, all right. Yeah, let's take a 5-minute bathroom break before we jump into the autonomic nervous system. So yeah, let's come back about 10:20 and we'll start back up. Yeah, the autonomic nervous system is one of the things in physiology where, if you kind of know in general what it does, what each side of it does, then it's not like you necessarily have to memorize every single little action, because it kind of makes sense. And so we'll kind of talk about that as we go forward. And I find that in a lot of things with physiology, it's that way. If you kind of understand the big picture, and in general, the way that the body works or what would make sense for it to do under certain conditions, then it can really decrease your memorization load, right? And so I always recommend for students to kind of take a step back with all of these things and like, all right, what's the big picture here? Like, what's actually going on? What is the body trying to do here, right? And then a lot of it can make logical sense, right? There's still going to be plenty of memorization there is, but it can cut down your memorization some if you can kind of rationally just like work your way through the problems to some extent. So this is a great example of that. So yeah, so we're kind of looking at our hierarchy here, right? We have the central nervous system, and then all the branches of the peripheral nervous system, the sensory, right? And then under the motor, we've got our somatic part, which would activate our skeletal muscle, but then our autonomic, which is controlling smooth muscle and glands. And so underneath that autonomic, we've got our sympathetic and parasympathetic. And so that's what we'll dive into a little bit deeper here. So you've all gotten the background information on all of this with the pre-class video. Let's just kind of walk through a little bit of comparing and contrasting between the two here. The first part would just be, where do the neurons come from? Okay, from this, where do they leave the central nervous system? Okay, so the sympathetic is going to be all from thoracic and lumbar nerves. Right, and then parasympathetic is going to be either all cranial nerves or sacral nerves. Right? And that's why it's called parasympathetic, right? Because it's alongside the sympathetic, it's on both sides of it. Sympathetic is all in the middle. Okay, so our general effects here would be, we've got our kind of fight or flight, right? So the actions of the sympathetic nervous system are either preparing our bodies to get out of there, right, as fast as possible, or getting out of there is not going to work. We got to fight, right? One of the two, right? Those are our two options with the sympathetic nervous system, right? So it's all about stress response, right? Okay. Whereas parasympathetic, we kind of just call it housekeeping functions. Right, so kind of just the general functions of digestion and urination and changing, focusing our eyes on things so we can see them clearly. Right, things that aren't necessarily immediately crucial, right? But they're obviously crucial, just not immediately crucial, right? And so one thing that differentiates the two is how long the effects of each system last. Right, so the sympathetic nervous system has long-lasting effects, and the parasympathetic is more short-lived or temporary effects. What's the main reason for that? Say it again, Ava. Okay. Yeah, that's some kind of practical reason. What would be the physiological reason? Because the sympathetic nervous system involves release of hormones too, right? Whereas the parasympathetic nervous system is all neurons releasing neurotransmitters, which can be, that activation can be removed quickly. When we send hormones out into the blood, we're just dependent on the half-life of those hormones, the breakdown of those hormones, right? So it takes a lot longer for those effects to die down, right? Um, effects is it's very diffuse in the sympathetic nervous system and very specific in the parasympathetic nervous system. Part of the diffuse thing is because of the hormone. Okay. Part of it is the branching. Zoom in a little bit. So there's a lot of branching of neurons in the sympathetic nervous system. So every, both parts of the autonomic nerve contain neurons or spinal cord to a ganglion, which is a group of synapses, right? Another close to the spinal cord, right? So then that postganglionic neuron is super long and it kind of branches actually just embedded in the target tissue, right? So we would have a long way to an organ like the bladder, and there's just a little short that connects to the neuron, just that very specific action, just right there in that tissue only, right? So so that length of the postganglionic neuron is very long, and these ones are very short. Okay, the other thing The other thing that's important to know here just is when we talk about the parasympathetic nervous system, or sorry, about the sympathetic nervous system, there's kind of multiple options on the pathway, right? There's always a preganglionic neuron and a postganglionic neuron with the parasympathetic side. On the sympathetic side, most of it is that way, but then the other option is you have a preganglionic neuron that goes to the adrenal medulla, right? So the adrenal gland on top of our kidneys, right? The inside of it, the very core of it, is a bunch of neurons, right? So it's nervous system tissue inside of our adrenal medulla, and that preganglionic neuron activates those neurons to release epinephrine and norepinephrine into the bloodstream, and those are the hormones that get released. So really, they're neurohormones, right? And so that's just another part of the sympathetic nervous system that's unique, and we don't have that kind of setup with the parasympathetic side. Okay, questions on that? Okay, so let's just talk about some specifics with each kind of organ that each of these would interact with. So with the parasympathetic side, looking specifically at the ones driven by our cranial nerves. Okay, so cranial nerve 3 would have its effects on the eyes, which would be constriction of our pupils. Right. As far as the glands go, we're going to have secretions in the eye. So we're talking about like our tear ducts and things like that. We're going to increase secretions in the eye. In the salivary glands, we're going to have very watery and profuse excretions, meaning that we're releasing a lot of fluid with parasympathetic activity. And that's the kind of big picture way to think about this is when we're not stressed out, we're free with water, right? We're like, yeah, we can spare the water to keep our eyes moist, right? Because we're not worried about that, okay? So I just said it's pretty much leading to secretions in the eye, leading to watery, profuse secretions in the salivary glands. Okay, effects on the heart. We're going to decrease heart rate. Okay, what do we think about in our lungs, in our bronchioles? Would we have constriction or dilation? When we're not stressed out. Constriction, yeah. Because we don't need to open up our lungs big if we're not really exercising, right? So we'd have bronchial constriction, okay? Digestive organs are going to be increased motility. Okay, so that's just like movement through the digestive tract. So motility, motility, motility. Okay, so that would be, oops, sorry. That would be contraction of smooth muscle. Okay, so we're thinking about like peristaltic contractions along our GI tract. We're going to contract that stuff to push things through, right? But then when it comes to sphincters, we're going to relax those with parasympathetic activity, right? Because that's what allows things to push through. So when we open up those doors, right, those sphincters. So relax sphincters, contract other smooth muscle around the lining of the tubes. Okay, with the kidneys, it's going to be increased urine production. Again, we're being free with water when we're not stressed out, so we can let go of more fluid. We can focus on things like getting that food through our GI tract, right? Because we're not stressed out. Okay, questions on any of those? Okay, then let's look at parasympathetic, the sacral nerves. Okay, so effects on the distal half of the large intestine are going to be the same as what we were just talking about, contraction. all right, which is going to squeeze down and push feces out. Okay, effects on ureters and the urinary bladder would be contraction as well. And effects on the penis and clitoris is erection. This is one part that seems a little bit of like an oversimplification of the parasympathetic side. We'll see that ejaculation is driven by the sympathetic nervous system and erection is driven by the parasympathetic. Okay, questions on these? Yeah, Ava? Uh-huh. It's pushing the urine out, right? Squeezing down on the urine, trying to push it out. So we'll contract the bladder, but then we'll have to relax the sphincter right here to let that stuff come out, right? So parasympathetic activity contracts the bladder, relaxes the sphincter. Yeah. Okay. Other questions on that? Yeah. go ahead.
There would be like, on the GA system at the bottom, we have vasodilation.
Yeah, vasodilation, which really starts with nitric oxide release, which is gonna cause vasodilation and engorgement of large venous cavities that lead to erection. Yeah, but that is controlled by parasympathetic neurons. Yeah. Mhm. Other questions? Yeah.
The lower, the large intestine, you
Uh-huh.
get lower contractility, just so you get more absorption.
In the large intestine?
Yeah.
The reason this says distal half is because I think the upper half, some of that is controlled by maybe some of the cranial nerves. I'm not sure why exactly, but I think that's why that we don't have a lot of absorption happening in the large intestines. Mostly all of our absorptions takes place in the small intestine. We absorb a lot of water and some vitamins in the large intestine, but everything slows down a bit when it gets to large intestines anyways. And so it's kind of where we're compacting and pulling the water out. So we don't have as rapid of movement anyways, but yeah, I'm not sure if that fully answers your question, but yeah. Yeah, go ahead, Hannah. Uh-huh.
Doesn't the sphincter muscle controlled by the parasympathetic?
Yeah, so so activation of the pupillary sphincter muscle causes constriction, right? So when we have a circular muscle like that, when we contract it, it squeezes down. And when we have a circular, yeah, so then with the pupillary dilator muscle, you're getting us into next week's class, which is fine. The pupillary dilator muscle is sympathetically controlled, and it's not a circular muscle. It's longitudinal, and it actually just pulls on that dilator or on the other muscle and kind of rips it open. So so when we're not under stress, when we want to constrict our pupils, we activate that pupillary sphincter muscle. And when we want to dilate our pupils, we just relax that pupillary sphincter muscle. We don't even use the sympathetic side unless we're under stress. This is talking about sphincters here in the digestive organs and urinary tract, but not the same as the circular muscles up in the eye. Yeah. Yeah. Good question. Other questions? Okay. Here we go. Okay, so now sympathetic. Okay. So let's start over here in the mouth. I'm going to zoom in a little bit for my purpose of my handwriting. So effects in the mouth, we've got thick secretions. And small amount of secretions. All right, so when we're stressed out, we are saving fluid, right? So our secretions, our salivary secretions get thicker and we don't release as much fluid. Okay, when we look at the heart, we're going to increase heart rate and we're going to increase contractility. Okay, heart beats faster and more forcefully. We'll talk about the mechanisms of those when we get to the cardiovascular system soon. Okay, sweat glands are going to activate. We've got our little erector pili muscles, which make our hairs stand up, right? We're going to contract those guys because we want to try to make ourselves look bigger than our whatever is trying to fight with us. Doesn't work so much for humans, but it does for animals. Makes those, you've seen those wolves or like coyotes lift their fur up and they look kind of scary when they do that, right? Humans, not so much, right? Um, sorry, you still need to see that part. Effects in the eyes. We've got dilation. Oops, sorry. That one's just kind of like, if I got some animal chasing me, I don't really care to like see the details of its face, right? I just want to see it, right? And so it's like the bigger that pupil can be, the more I can see, right? I don't really care if I can see it clearly, right? So that's the way I think about that one. Effects in the nose would be, again, kind of thick secretions, right? And small amount of secretions. Okay. Effects in blood vessels. What do we want to do? We want to increase our blood pressure or decrease it when we're stressed out? Increase, right? So we're going to vasoconstrict. It's going to increase blood pressure. And then our bronchioles are going to dilate. Right? So we can increase our ventilation and the amount of air going in and out of our lungs. Okay. Questions on that one? All right, then we'll go lastly here, sympathetic effects on the lower kind of region of our body. So bladder is going to relax. Right? It's not a priority to urinate when you're under stress, right? It could be though that if your bladder's very full and you all of a sudden increase a bunch of pressure around it, you could pee your pants, right? So people always ask that question, like, well, don't some people like pee or poop their pants when they're really scared, right? If you have a lot of pressure in that space already, and then you create more pressure by clamping everything down, then there's a mechanical thing going on there that's different from this. And so on the male genitalia, as I already mentioned, ejaculation. Okay, adrenal medulla is going to be release epinephrine, and norepinephrine. Effects on the liver going to lead to glycogenolysis, which is breaking down that stored carbohydrates, and so we can increase our blood sugar, right? Effects on the pancreas, we're going to decrease digestive enzyme release, right? Digestion is not the priority. Effects on the large intestine, we would relax most of the smooth muscle because we're not trying to move things through at that point, but we would contract our sphincters to stop movement through the digestive tract. And this is just a selection of things, right? There's obviously a lot more with all of these, right? We just kind of picked some for each kind of region and organ. But yeah. Okay, questions about those? Yeah, go ahead. Yeah, for sure. Definitely related to constipation. Absolutely. Mm-hmm. Yeah, go ahead. Hmm. Yeah. Yeah, I think part of that is just a fatigue thing, right? Like where the muscles that are holding everything in, your sphincter muscles, are just gonna eventually kind of wear out. Part of it too can be a pressure thing when we get to the digestive tract. I don't remember the numbers off the top of my head, but and we'll talk about sphincters. We have the internal anal sphincter and the external anal sphincter. The internal one is controlled by reflexes. The external one is voluntary skeletal muscle that we can control. But at a certain pressure on that sphincter, we can't control it anymore. Reflex takes over and opens it up. And so that could be part of it too there. Yeah. So yeah. Okay. Okay, so what we're going to do next, this is just kind of a cool chart. You don't have to memorize all of this by any means, but many of the drugs that you'll prescribe someday are affecting the autonomic nervous system. Right? There's just kind of a cool image that shows different branches of this. Okay, so the kind of the takeaways to understand here are, so first of all, these are all sympathetic drugs, right? Drugs that dealing with the sympathetic nervous system. Anytime we see alpha or beta, that's only going to be referring to adrenergic receptors. Okay, adrenergic receptors, think of adrenaline, right? Adrenaline is part of the sympathetic nervous system. Okay, so if we see alpha or beta or we see adrenergic, that means it's acting on the sympathetic nervous system. Okay, if we see cholinergic, that means it's talking about acetylcholine. Okay. And that means it's going to be on the parasympathetic side. Okay, if we see muscarinic or nicotinic, those only refer to cholinergic receptors. So then again, that has to be a parasympathetic drug. Yeah, go ahead. Yeah, for sure. Yeah, and we'll do some questions that'll help with this too. But adrenergic is always sympathetic, right? If you see beta or alpha with any number, that's only referring to adrenergic receptors. So that also has to be sympathetic. Okay. If we see cholinergic, that's referring to acetylcholine receptors. That has to be parasympathetic because this is all talking about the actions of these systems on the target tissue, right? So it's always adrenergic receptors with the sympathetic nervous system, and it's always cholinergic receptors on the parasympathetic side. We never would use muscarinic or nicotinic to be talking about the sympathetic because muscarinic and nicotinic receptors are types of cholinergic receptors. So cholinergic, muscarinic, nicotinic, all referring to the parasympathetic side only. Okay, and then the last ones to remember would be whether it's an agonist or an antagonist, right? So if it's an agonist, it does what that thing would do normally, right? If it's an antagonist, it blocks what that thing would do normally. Okay, there's a couple other cool terms down here. We talked about this one already. Sympathomimetic, right? That mimics the effects of the sympathetic nervous system, or sympatholytics, which would be blocking the effects of the sympathetic nervous system. Okay, so what the way that I'll kind of ask questions on this will be more about giving you the information about the drug, like saying the drug does this, it's a beta adrenergic blocker, right, or beta adrenergic antagonist. So then what would its effects be on this certain tissue? Okay, so you don't have to memorize what the drug does yet for this class, okay, but you have to understand by that terminology and then by your knowledge of what that system would do to that organ, what the effect would be. Okay, so we're going to do a bunch, 10 questions right now related to that exactly. 5 or 6 of them are match these slides, but then I added a few more. Okay, so yeah, let's go ahead and switch over to that. Okay, let me make sure the camera's still working. I don't think it is. There we go. Nope. Yeah. That's why it turns it off for you automatically. But the endless thing. There we go. Okay, let me make this QR code a little bit bigger so you can see it. Go ahead and hop on there with your phones or iPads, whatever you want to do. Yeah. Oh, it went away. I did make it bigger, then it went away. Sorry. Thank you. Oops. There we go. Sorry. Wait. Oh, I have to hold it or something? Maybe? That's weird. Nope. It lasts for like 2 seconds. I don't know why it does. Yeah. That's not helpful. You can just type that in, I guess. If you can't get the QR code to work, I don't know why it doesn't stay for longer. Maybe if I hold it, nope. Can you scan it when it's like that? Yeah, never mind. Sorry. All right, most of you seem to be getting in there.
Yeah.
All right, how many we got? 29. There we go. Here we can time it. Oh, is it staying? There we go. Maybe I was messing it up somehow. Okay, there it is. Nope, then it went away. Okay. All right. Anybody still trying to get on there? Okay. All right. So let's go ahead and start. It'd be great if you kind of chatted with your partner with each of these, and then sent your answer. So let's see here. Is everybody in there? Yeah. Okay. Okay. You guys already started some. Yeah. Here we go. Okay. So if a drug, here we go. Nicotinic, muscarinic. What does that mean? Go ahead and chat with your buddy just to make sure you're on the same page. See what you guys are putting here. Okay. Why can't I see? All right. Yeah. So most of you are saying the parasympathetic nervous system. So nicotinic, muscarinic, those only refer to subtypes of cholinergic receptors. Cholinergic receptors are acetylcholine acts on them, right? And that is always the neurotransmitter at the target of parasympathetic neurons. Okay. So parasympathetic would be the way. What else would be, what other ones would be true? Autonomic and peripheral. Yeah, for sure. Yeah. And one more? Efferent. Yeah. Yeah, it's part of our motor side of our system, right? Not autonomic nervous system. Okay. All right, let's go to number 2. You guys are already answering a lot of them. This is a choose all that apply. It does? Oh, okay. Never mind then. Tried to create that. I made multiple answers correct, so but you can still only choose one. Choose your favorite one then. I don't know why I can't see how many of you guys have responded this time. Last time I could. What's that? At all, or just the multiple answer things not working? Okay. Huh? Yeah, so we good. So we've got if a drug acts on alpha or beta or alpha receptors, which of the following are true about that drug? Is it parasympathetic or sympathetic? Sympathetic, got that part. Cholinergic? Nope. Adrenergic? Yes, for sure. Adrenergic, always going to be norepinephrine or epinephrine. Okay. Couldn't be muscarinic. could be a sympathomimetic, for sure. It could be mimicking the effects of the sympathetic nervous system. It could also be blocking the effects of the sympathetic nervous system, which would be this one, sympatholytic. Okay? Could not be a nicotinic, could not act on nicotinic receptors because those would be on the cholinergic side only. Okay. All right, next one. Yeah, and it looks like you guys can just go through and answer these as we're going. Okay, atropine is a muscarinic blocker. We could also call it a muscarinic antagonist. So when atropine drops are placed in someone's eye, what would happen? The pupil would dilate because we're blocking the effects of the parasympathetic nervous system, right? So it's parasympathetic. Activation of the parasympathetic causes constriction. So if we're blocking that, we do the opposite of that. We dilate instead. Okay, questions on that? Okay, all right, next one. All right, phenylephrine is also used by optometrists for examining the eye. It's a sympathomimetic. So what effect would it have? Talk to each other. Make sure you're on the same page. Talk about what we haven't talked about those muscles yet, besides with Hannah's question. um, all right, we'll get into the those muscles in more detail next week, but we've got the pupillary dilator muscle, which is the one what's controlled by the sympathetic nervous system, and when it's activated, it pulls open your pupil. Some of you may have done in a lab in undergrad where you pinch the back of somebody's neck while you're looking at their pupils, and their pupils will open up like that real quickly because the pain response to get that sympathetic activity. You could try that on each other if you want. You got to get pretty close in their face to see it happen, but you can kind of see, do that little pinch and their pupil opens up really quick, right? It's that sympathetic activation of the dilator muscle. Okay. Next, albuterol is a beta-2 agonist. Okay. So most of you are saying albuterol causes bronchodilation, right? Because the actions of the sympathetic nervous system on our lungs are to open up our airways, right? So we can breathe more. So if this is kind of mimicking that effect, right? So we're activating the sympathetic nervous system here. So this would be a bronchodilator. Sure. Okay. All right. Let's go to the next one. Metoprolol, the beta-1 antagonist. Okay. So what do you guys think? Talk to each other about this one. Ask each other. All right. So if we got a beta-1, what side of the autonomic nervous system are we talking about? Sympathetic. And if it's an antagonist, are we doing the same as the sympathetic would do or the opposite? Opposite. Perfect. So we know the sympathetic increases heart rate and increases contractility. So if we're blocking those effects, we're going to do the opposite. We're going to decrease heart rate and not increase contractility, but decrease contractility. Okay. Questions on that? Okay. Next one, phenylephrine. Again, alpha-1 agonist that affects blood vessels. So what would it do to our blood pressure? Most of you are saying it's going to cause contraction or vasoconstriction of the smooth muscle around our blood vessels. And by doing that, that increases our blood pressure. So this one is correct here. Okay. Ready to go to the next one? Okay. All right. So propanthyline is a muscarinic antagonist. Okay. You guys are all over the place on this one. So talk to each other, see what you think.
That's great. I'm making more sympathetic. Yeah. More E, one A.
So the effects on the bladder and the ureter specifically, not the sphincters. All right, so propanthyline is used for urinary incontinence, okay? And so the correct answer here would be we want to relax the bladder and the ureters because if we contract them, that pushes things out, right? So muscarinic antagonist, right? And so we're blocking, so muscarinic is which side? Parasympathetic. Antagonist would be doing the opposite of the parasympathetic, right? So parasympathetic normally contracts these so we can urinate, right? This is doing the opposite of that. We're relaxing those so we don't urinate, right? Okay. All right. This one, same drug, but what about its effects on the sphincters instead? Check with each other on that. Yeah, good. So everybody's saying the same thing. This would lead to contraction of those sphincters, which helps with urinary incontinence, right? Because we're closing that door, not letting the urine out. Okay, and then lastly, yeah, this is an interesting one. Neostigmine, which is an acetylcholine esterase inhibitor. So a different level of drug here. It's not acting right on the receptor. It's acting on the enzyme used to break down acetylcholine. It can be used in cases of severely delayed gastric emptying. How would that work? Talk to each other about this one. This is our last one, so we're just about done. All right, so what side of the nervous system are we talking about here? Parasympathetic is the cholinergic. Synapse we're talking about, okay? The main way that we get rid of acetylcholine after it's been released, after an action potential arrives, is by this enzyme that breaks it down, acetylcholine esterase, okay? And so these drugs have been discovered, and it really started with like a bean that had this stuff in it, and then they make it synthetically now. But pretty much, if we're blocking acetylcholine esterase, that means we'll have more acetylcholine out there, right? Which means we'll have more of the effects of the parasympathetic nervous system, okay? So most of you are saying it causes inhibition of the breakdown of acetylcholine in smooth muscle, the GI tract, which would lead to contraction and increased motility. True, that's the one. Okay? All right, yeah, go ahead.
Drugs like just target one system.
Yeah, it's a great question. I haven't looked into it enough. This was one I just found about 5 minutes before class as I was making this one, but I just thought it was interesting. But yeah, it's a good question because yeah, you would think like this could affect your skeletal muscles, could affect all kinds of things. Yeah, I'll look that up to see. It's really interesting for sure. Was there another question? Okay. Hopefully you're seeing a little bit with this autonomic nervous system what I was talking about, that you can use logic to get your way through it in a lot of cases. Okay, so try to step back and look at that big picture. But all right, guys, we're done. Have a great rest of your week. We'll see you next week. All right.